The article is about genome edited crops. So if we see, cultivation of GM crops, it is only for cotton in India. So there is no technology approved for commercialization since Monsanto's Polgar 2 BT cotton in May 2006. In May, two genome edited rice lines identified for research, and it improved genetic edited mutants. Number one, Samba Mansuri, and it led to 10% yield increase. Number two, MTU 1010, and it can grow in saline and alkaline soil. Now if we talk about third genome edited line in mustard, so it's cannula quality low pungent mustard, and it is resistant to fungal pathogen and pest, and it will release in August 2006. So if we differentiate between genome edited and genetic modified crops, so first let's talk about genetic modified crops. It contains foreign genes from unrelated species. For example, Bt soil bacterium, it codes for production of proteins which is toxic to various insects pests in cotton. Now if we talk about genome edited crops, uh, in genome edited crops, it editing editing of genes occurred naturally, which is present in host plant, and it led to mutation and change in DNA sequence. And editing is done by protein scissors. It acts like molecular scissors, to cut and modify DNA of targeted native genes. Now, such editing aimed to bring desirable alternatives. So, uh, an enzyme is released, and it enzyme is directed to a specific location oblique address in plant genome by a navigator guide RNA. So what is navigator guide RNA? It custom designed molecule having sequence matching that of targeted DNA. So if we see some examples of genome edited crops, for example G rice and mustard, so scientists used CRISPR associated Cas9 protein, and Cas9 protein deployed editing drought and salt tolerance gene in MTU 1010 rice, number one, and it deployed in editing 10 glucosinolate transporter gene in Varna mustard. Number three, Cas9 protein deployed in editing cytokinin oxidase 2 GN1A gene in Sama Mansuri rice. So Cas9 in GN1A gene, Cas9 codes for enzyme that degrades cytokinin. Okay, cytokinin is actually a plant hormone that is stimulating formation of rice, spiculates or flowers. So more cytokinin means greater number of spiculates that develop into grains. So by reducing expression of GN1A gene, degradation of cytokinin can be lowered. So increased number of grains, panicles, and yield. Now, what is the policy push for genome edited crops? Number one, genome edited plants are free from exogenous introduced DNA, and it has been exempted from Ministry of Environment and Forest and Climate Change. Okay. Number two, improve two major rice varieties, which has been initiated in 2018 through National Agricultural Science Fund, and 500 crore were budget outlay in 2023-2024 for genome edited crops, 310 crore for field crops, 40 crore for animal science, and 20 crore for fisheries, and 10 crore for edited microorganisms. And it identified 178 genes in 24 field crops for editing, and 23 targeted genes in 16 horticultural crops. Now, if we see human resource development in genome edited crops, 9 scientists were sent for advanced training in US, Europe, and Australia, and 10 scientists from Innovative Genomics Institute. Okay. Now, a team led by Kutubuddin Ali Maulia presented an indigenous miniature alternative G tools that uses TNPB or transposon associated proteins which sourced from another bacteria. It is cheaper because IPR is with India, and it is more effective because protein size is smaller than Cas9 and Cas12a.